Today we shall discuss the structure of microsporangia. A typical anther of angiosperm is bilobed with four-sided, that is tetragonal, and each side contains a microsporangium, and each side contains a microsporangium. This is lobe 1, this is lobe 2, 1, 2, 3, 4 sides, and contain four sporangia. This is four sporangia, and in this separation, this is a sterile tissue to separate the lobe, and this is microsporangium. Now this wall, you see the anther wall from here to here. This is anther wall. It is composed of, means from here to here, it is anther wall, and this anther wall is composed of anther wall is multi-layered and contain four types of cells lining. Contain the outer wasp. This is this one. This is the outer one, that is first the outermost is epidermis. It is single layer. It is single layer, flat cells, and their main function is protection. Their main function is protection of the anther. Below that, this is the next layering of the cell. It is also single layer, and this is known as endothecium. It is also single layer. Cells are compactly arranged. Cells are radial and compactly arranged. Cells are, this one is, and in between these there is, in between these two, a callous layer is found, which joins the, which binds the spore tetrad. So there is a lining, and this callus is found between that. So the far, and below that there is 3 to 4 layer and now turn on. Middle layer, that is 3 to 4 layers. They are loosely arranged. They are loosely arranged, made of parenchymatous cells. So these three means the outermost epidermis, next one endothecium, and third one middle layer. They all help in the dehiscent of the anther. Now the fourth layer is of tapetum. Now the fourth layer is the tapetum. It is the most important layer. That is tapetum. Pyramid shaped, usually multinucleate. In some cases, the uninucleate is also found. If the uninucleate, the nucleus is big in size. It is a nutritive layer. Yeah. It supplies food to the developing pollen grain. It is also important because it contains enzyme callage, which is essential at the time of separating spore tetrad. It also contains ubisque bodies and also help in the formation of sporopollenin. It also helps in the formation of sporopollenin. It is a hard resistance compound and it is found on the wall of the pollen grain, that is outer wall exine. So this is the function of tapetum. And now in the middle of this sporangium, this one in the middle of the tapetum, this is sporangium is found, means inside the tapetum layer, the microsporangium is present, containing compactly packed sporogenous cells. In this, the compactly packed this is sporogenous cells, that is compactly packed. So it is a fourth layer. Outer one epidermis, single layer. Next one endothelium, it is also single layer. Third is middle layer, that is 3 to 4 layer, and finally the tapetum, the nutritive layer, is found. And inside the tapetum, the microsporangium is found, and which contain the sporogenous cells. Microsporogenesis, that is formation of microspores, that is formation of male gametophytes. It is also called male gametophytes. Now this microsporangium cells, means as the anther develops, the sporogenous cells get loose, and each sporogenous cells acts as microspore mother cells, which is diploid, means that is 2n. Now this microspore, these are microspore cells. This is microspore mother cells. They undergoes meiosis. And each cell forms that is spore tetrad, 1, 2, 3 and 4 on the back side. So this is for spore tetrad in 4 cells, and this is haploid in nature. This is haploid. This spore tetrad may be of following type. It is either of this type or of this type. This is known as tetrahedral type. This is known as isobilateral type. and this is known as linear type, and also an F separate decussate type is also found. So these are the different types of sporotractors, but this one is more common in angiosperms. Now this microspores is found, and it is the development of male gametophytes, and this microspore now undergoes development, and these tetrads, and they separate from each other and change into the microspore. Now they separate in each other, and this is called microspores. So these are the microscopes, and they develop into the male gametophyte. Now these microscopes will develop into, now it develops into pollen grain. So here you see that that in this, the next structure, you see the cells in undergo spore tetrad is found in that. So after the next stage, the meiosis, the spore tetrad is formed, and now they get separate. Now that is, this is microspore mother cells, and this is 2n. Now it undergoes meiosis and forms a spore tetrad, that is haploid n. Now these cells get separated, and these are known as microspores. These microscopes develops into pollen grain. This is the microscope, this is the plasma membrane of that, and around the microspores, a wall intine and exine. This is the exine, that is outer wall, and this is the intine, that is inner wall. So these are developed in that, and in such condition it is called pollen grain, means microspores. When it covers under the exine and intines, then it is called pollen grain. Intine made of cellulose plus pectin, and exine, you know, that is, it is made of sporopollenin, that is highly resistant substance, and by which it can resist, it can survive in the highly temperature, and also there is no effect of enzymes on that. It contains a single nucleus. Now the nucleus undergoes mitotic cell divisions. And gives us two cells. Understand? And in that nucleus, now the nucleus undergo mitotic division, two nuclei are formed, and there is cytokinesis. The cytoplasmic division is unequal, so a large cell and a small separate cell is formed, and in that there is also a structure is found that is known as germ pore. Here X sign is absent, and these two cells, the bigger one is known as vegetative cell and the smaller one is known as generative cells. It is also known as tube cell. So this divides into two. This is the germ pore for the growth of the tube cells. Now in such conditions, mean two celled stage, it is come out of the anther. Means as one thing you know, this is the anther wall, and this one is called point of descent. This is called point of descent. These parts all are fibrous in nature except that point. Except the point of descent, the other parts are fibrous in nature. And when this pollen grains is completely formed, suppose this is the complete pollen grains is formed. Complete pollen grain is formed in the fourth stage. Understand? And now it will come there. And after the formation, the anther will burst from the point of descent, and this pollen will split, and these pollen grains in two celled pollen grains, two cells pollen grain will come out. In the majority of cases, the two-celled pollen grains are come out. One of vegetative cells and next one is generative cells. Understand? But in some cases, the three-celled pollen grains also come out. In such cases, in the three cells, the generative cells also divides into two male nuclei. So in that way, it becomes three-celled. So either through two cells or three cells, it comes out of the anther. Now it goes to the stigma by the process of pollination. In that way, the male gametophyte develops and it undergoes the next process.